So at this point, we shift to the President's Plenary session, which is called Taking Stock of the Connectome. As many of you know from bitter experience, the brain is complicated. And in our 51st year, the ACNP still struggles to deliver on the promise that our college really made at the time of its founding to use neuroscience to develop new treatments to alleviate the suffering and disability associated with neuropsychiatric disorders. So as I was thinking about the topic for the session today, the plenary, it seemed to me that the human connectome was at the core of our current translational neuroscience efforts and the effort to define neuroimaging phenotypes that might guide diagnostics and therapeutics. So today's panel is an effort to take stock of the pursuit of the human connectome and to consider the opportunities and challenges associated with this effort. This panel will also consider the issues of how to use this connectome productively, how to integrate it with other kinds of translational neuroscience approaches such as genetics, and how understanding of the connectome may inform drug development and other critical processes in the clinical world ultimately. So our first speaker today is Dr. Def Lippmann. And I wanted to begin this session, this plenary, with a presentation that would help us to understand the true complexity of the development of connections in the brain. Dr. Lichtman's presentation will provide a visually, I assure you, awe-inspiring insight into the complexity of the development of connectivity and make us very humble and cautious about over-interpreting what we are learning from functional neuroimaging. Dr. Lichtman is professor of molecular and cellular biology at Harvard University, and his research yields a unique record of the way that the formation and elimination of synapses in the developing brain, particularly in the context of life experiences, yields distinct measures of complexity at the micro level that serve in a way to encode experience throughout life. So it's my tremendous pleasure to introduce our first plenary speaker, Dr. Def Lippmann, who will be speaking on order and complexity in the development of synaptic connectivity.
Thank you, John. I'm very grateful for this opportunity to speak. Although I have an MD degree, I think most of what I'm going to say is far removed from useful clinical information. I'd like to begin by pointing out something that I'm sure most of you are aware of, at least subliminally, and that is for almost all organ systems in the body, a central way we've learned how they work is by understanding the relation between their structure and function. And indeed, this has been a remarkably powerful way to understand not only the normal function of organs, but also their pathologies. And many of you probably appreciate, if you went to medical school or took a histology course, that for each organ system, except for one particular, there is a kind of cellular motif. And once you understand this cellular motif, you basically have a good sense of how that organ works. In the kidney, it's a tubule system. In the liver, it's a portal triad. In the lungs, it's the place where vessels of air, bronchioles, come in contact with blood vessels for gas exchange. Once these cellular motifs are understood, the function of the organ is pretty well understood because that little cellular motif is iterated over and over and over again, making it pretty straightforward to understand the normal organ. And with very few exceptions, diseases of these organs, except for one particular organ, have very obvious manifest changes in the pathology of this cellular motif. For some reason, this has worked out very well for every organ system except for the nervous system, where, as you all know, there are a wide range of disorders of the nervous system where there's no doubt, looking from the outside, that the behavior of an animal or a person is deranged. And yet when we look at the nervous system, we find it very difficult to see what actually is wrong. And trying to fix something that's broken where all you know is that it's not working right, but you don't actually see what's wrong, it is a tremendous challenge. And so I'd like to begin by arguing a little bit about why it might be compared to other organ systems. The relationship between the structure and the function of the nervous system is much more complicated. The first reason for this is that there has been an astounding increase in the complexity of the nervous system compared to other organs. Whereas there are maybe 5 or 6 cell types in a kidney, there are countless numbers of different kinds of cells in the nervous system. A state-of-the-art question right now, for example, is to figure out how many different kinds of cells are in the retina. The retina is the most well-organized of all central nervous system tissue, and to this day we do not know, and in fact many people are trying to figure out exactly how many kinds of cells, not the total number, but just different kinds of cells are in the retina. And that number will be somewhere between 40 and 400, I guess, and someday we will know how many cells there are in the retina. But this isn't going to help us very much to know how many cell types there are in the cerebral cortex, or in the brainstem, or in the subcortical nuclei, or in the spinal cord. There is this just remarkable structural diversity, and going along with this is this extraordinary functional diversity. You could write a chapter, maybe 100 pages or so, to say everything a kidney does, but what about for the brain? If you had to write a chapter on everything a brain does, you couldn't write that chapter, for humans especially. It's an open book because we keep doing new things with our brains, things that were not encoded in any obvious way because the tools we're using are changing so rapidly. So this is one of the challenges of understanding the relation between the structure and function of the brain. There's a second problem, and that is compared to other organ systems where the same idea is iterated over and over again. So if you understand a little piece of lung, you basically understand the entire lung. In the brain, there is organization at many different size scales. And I'm sure all of you are fully aware of this, that if you look at the brain over a cross-section of the entire brain, there's a division of labor so that planning for the future may be near the front, vision is near the back, balance is in the cerebellum, the brainstem has a lot to do with things like breathing, and the spinal cord is essential for other features. If I take out one of your kidneys, you still behave pretty normally. If I take out one of your lungs, you still can survive. But if I take out half of your brain, there's no question you'll have a big effect. And that's because every part of the brain has unique functions. I can take out small regions. Even with strokes, you see remarkable changes. So at one end of the spectrum, one has to understand the brain at this very macroscopic level. And you know, the Fmri technique, where the voxel size is about a cubic millimeter, gives one sense of division of labor at this level. But as one delves deeper and deeper, one sees division of labor even in the cortex itself, the white matter and the gray matter. Even individual neurons have a division of labor. They have apical and basal dendrites and an axon that can course over to the other hemisphere and to the spinal cord. One zooms up on one of these dendrites of an excitatory neuron in the cortex of pyramidal cell, you see dendritic spines. And if you want to actually visualize what's going on at the spine level, you have to go to the electron microscope and see synaptic terminals filled with vesicles on spine apparatus. This is an order of magnitude of 6 to get from the big to the small. And just to give you a sense of how huge a problem this is, an fMRI voxel, which is a cubic millimeter, is 1 trillion times larger than the full resolution you would need to see synapses, which would be 100 cubic nanometers. So there's a 1 trillion-fold range in resolution one has to deal with to understand the brain. And it's not just the big and the small, that's everything in between that also has to be understood. This makes the brain a much harder organ to understand than any others. And these are the easy problems, because there's a third problem that is really difficult, especially for human brains and the brains of other mammals. And that, like other animals, we start with the idea that the brain its function comes from its structure, and its structure ultimately comes from a genetic book of instructions. That's true for all organ systems, but the brain has one extra feature, and any of you who learned to ride a bicycle as a child appreciate this, and that is during experience you learn things, and those things last for a lifetime. Is there anyone here who did not learn to ride a bicycle as a child? My neighbor is a woman who grew up in Africa, and she moved to the United States, and she has 2 children who are in their one's 8 years old and one's 10. They're adolescents, and they're learning to ride bicycles very easily, and they're beginning to ride around the block, and this woman, I think, has become a little nervous that they're getting out of her field of view, so she decided she would learn to ride a bicycle as well. She had not learned as a child, and I watched her last summer learn, or I should say attempt to learn to ride a bicycle. She is, as far as I can tell, neurologically normal in all respects, except when she gets on a bicycle, where it seems like her cerebellum is missing. You know, her arms are going back and forth like crazy. She can't seem to balance. And the striking thing is that I know that although I learned to ride a bicycle as a child, I spent about 30 years of my life in St. Louis, and I did not ride a bicycle when I was there at all. I did most things by walking or by car, and then I moved back east, and my wife noticed that I was beginning to grow, but not up, but out, and she suggested that we exercise and maybe get bicycles. So we got bicycles, and I hadn't been on one in many decades. And about 8 seconds after being on the bicycle, I was fine. So somehow I had something in my brain that my neighbor didn't have, and I don't think I had a gene for bicycle riding. What I had was the experience of riding a bicycle as a child, and that experience had changed me in a way that even without practice, I held on to this. So you can think of function in that gene structure function. Function in this case being experience and practice has generated a change in the structure, which then gives me a new function. And that loop, where there's no cause and effect, where function generates structure and structure modifies function, is a fundamental part of the nervous system, and it means that the structure of the nervous system is going to be complicated in a way that other organs are not complicated. Indeed, if you think about memory, the formation of memory, the old term that Richard Semon came up with, the engram, you might think of this loop as the process of engrammatization. I made up that word, but that is a very interesting part of the nervous system, and indeed that is why I study the wiring diagram of the nervous system. Now, until recently, we have gotten most of our insights into the structure of the circuits of the nervous system by taking advantage of sparse labeling techniques, and the first sparse labeling technique I'm sure you're all aware of is the Golgi Stain, which allowed Ramon Cajal to posit that nerve cells are connected in a remarkably interesting directional way. And here's a classic example of his drawings where the paper has yellowed, the whiteout he used to correct has stood the test of time. So that's not snow on trees here, that's just whiteout. But what I'd like you to notice is down in the left-hand corner at E, there's an arrow ascending, and that is an axon that's making synapses on the dendrites of a neuron, and that neuron then collects that information, then conveys it out its own axon, which hits the apical dendrites of another cell, and that information is conveyed into that cell along with the inputs from another axon on the more proximal dendrites, and according to Cajal, those inputs collaborate in some ways to generate a signal that leaves that cell out its axon, and that axon innervates the dendrites of other cells, and each of those cells then send information out their axons. This was a profound insight, and it has, to a greater or lesser degree, stood the test of time, and it was so simple that virtually anyone could understand it, and it has, I think, commandeered our way of thinking about central circuits. I'll give you a good example. That picture was drawn in 1913. Here's a picture from Rudolfo Llinas, a giant in cortex studies, and colleagues in 2002. And this drawing has many of the same properties as that original drawing, except that those green cells are inhibitory, and Cajal did not know about inhibitory neurons. This sparse worldview is so dominant in our thinking that very few of us can think about cortical circuits without thinking about these arrowed wiring diagrams. In fact, we have nothing else except in one animal, the C. elegans, a worm that has 300 nerve cells, and in that animal, every connection has been mapped. I'm just going to show you a picture of that wiring diagram. This is a saturated worldview as opposed to a sparse one. The sensory neurons are at the top, the motor neurons are at the bottom, and the few interneurons are in between. And the disturbing thing about this is that you can almost get from anywhere to anywhere in this wiring diagram, and there's no point putting arrows in it because no one would know exactly what direction those arrows should go. And yet this is closer to reality, I would argue, than the Cahalian view that we've all grown up with and accept. I think this equivalent in the mammal is worth considering. It might be in some ways simpler, even though there'd be more cells, than the wiring diagram in a worm, because in the wiring diagram in the worm, everything is based on evolution. Every single cell is a special case. There's no redundancy as there are in our nervous system. For many reasons, therefore, we've been trying to do the same kind of thing in the mammalian nervous system. And mapping all the connections is like genomics is mapping all the genes. This would be connectomics. And here's the dictionary definition if you hadn't heard of this term before. It's pronounced connectomics or connectomics. It's a plural noun, but singular in construction. This, by the way, in the small print there is from Merriam-Webster's Unabridged Dictionary 2015, maybe. A branch of biotechnology concerned with applying the techniques of computer-assisted image acquisition and analysis to the structural mapping of sets of neural circuits or to the complete nervous system of selected organisms, using high-speed methods with organizing the results in databases, and by this I do not mean Excel spreadsheets. This is a different kind of database than most biologists are used to, and with applications of the data in neurology, psychiatry, fundamental neuroscience. You could imagine that there are diseases of connections, connectopathies, if you will, where the problem in the brain is what's connected to what is abnormal. I don't think we're going to have the proximate cause for these diseases until we have ways of looking at the connectome at this level. And I stole this language by looking at definitions for words like proteomics and genomics, and the word connectome might be the full, complete wiring diagram of the nervous system. So how to do this? One possibility would be to go back to the Golgi stain, but rather than do it with one color and sparse, do it dense, that is, get everything labeled, but have every cell a different color. And thanks to the revolution in fluorescent proteins that some of you probably are aware of, the green fluorescent protein from jellyfish and now a bunch of mutations and other marine animals give us proteins that come from genes that generate fluorescent colors. And the spectral diversity provides an avenue forward. And one approach might be to take advantage of green fluorescent protein genes, red fluorescent protein genes, and blue fluorescent protein genes. RGB is really all you need to get all the colors a human can see because we are, after all, trichromic. That is, every color we see is just a mixture of those three colors, including the colors on the screen, which are just three channels being shown there. And so that's how black and white became color television, with just three colors. So if one could randomize the amount of red, green, and blue fluorescent protein in individual cells, that might be a strategy forward. And indeed, Jean Levet, who was a postdoc at the time, he now has his own lab in Paris, and Josh Sains and I built these constructs that generated multiple colors. This is a DNA construct that has three genes in it, one for red, one for green, and one for blue fluorescent protein that's expressed in the nervous system. And in the presence of a particular enzyme called Cre, the construct has to choose whether it's going to express red, or following recombination, it's either going to express green or blue. So if you make an animal with that construct, not surprisingly, you get a brain where cells are one color or another. This particular line of mice we made, in this case, has three colors only in astrocytes. So this is the the paintball equivalent of what astrocytes look like in the brain if everyone is randomly colored in red, green, or blue. And so we call these animals brain bow, so this would be astrocyte bow. If I zoom up on this, you can see what's wrong with 3 colors. It's not enough colors to really show you every single cell. This blue cell here is probably about the size of a typical astrocyte. These yellow cells are different cells, but with 3 colors, you don't have enough colors to see everything. It turns out, however, that when you make transgenic mice that have a construct like this, often multiple copies are in tandem, one next to another, and each of them will randomly choose, in the presence of Cre, one of those 3 colors. So you might have one cell that has 3 doses of red fluorescent protein, 1 of green and 1 of blue. You add all that up and the cell would be a brownish orange. Another cell might have 3 greens, a red and a blue, and end up being a spearmint color. And these brains have many more colors. The cortex is at the top and the hippocampus is right in the middle there. And here's the dentate gyrus. There's clearly a lot more colors in these animals. We've used these to watch axons that grow out to the periphery. Here's an example of these rainbow mice. This is a new generation of them that are brighter than the original ones. Each axon has its own particular color, and that allows you to follow them over very long distances. We did not discover this idea. This idea was first invented by computer scientists trying to figure out how thinking machines work. But it's the same idea, and when you trace these axons out to the periphery and look in a muscle, you see the neuromuscular junctions, and the color of the synapses, these pretzel-shaped objects, are the neuromuscular junctions on invisible cylinders of muscle fibers running up and down. You can say with certainty which muscle fibers are innervated by which axon. So you see there are 2 purple neuromuscular junctions right in the middle there. They're innervated by the same nerve cell. Even though the cell is very far away, I can still tell locally that they're the same, and I can also say with certainty that virtually none of the ones around it are the same axon. In fact, every axon there is almost a different color. Using this, one can take a small muscle, like the interscutularis, and Julu did this, a graduate student at the time. That's just a joke. And then he generated the entire wiring diagram of this muscle. Interesting thing about this wiring diagram is that every axon more or less went everywhere in the muscle, but each neuromuscular junction was singly innervated. Each of these colors is one of those things I was rotating about a field about this big from the previous picture. And the first question we asked is this a stereotyped wiring diagram? Will it look the same in one instantiation after the next? And we compared the left interscutularis muscle, which wiggles the ears, with the right interscutularis muscle. As you know, mice are bilaterally symmetric, so this was an easy question to ask. And we found that every single instantiation was completely unique and wired differently. Even in the same animal, the left and right were wired differently, but the left of one animal was wired differently from the left of another, so every instantiation was unique. But even more surprising, perhaps, was that there was lots of suboptimality in this wiring. A lot of useless loops, premature branching. Here's an example of an axon in red. I'm just going to zoom in on this part. All the axons were labeled, but I'm just showing one here. All the axons go this way. This axon and one other decide to go this way. The one that's slightly in blue went both ways, but this red axon decided not to go this way, which is its preference in this particular animal, and it goes all the way up here, and then for some reason makes a hairpin turn, goes backwards, and goes down here. If it goes any further, it's going to leave the muscle, and then it makes a hairpin turn and go out again. This little branch here goes to this neuromuscular junction all the way down here. The fast way to get there would have been just to go this way, but no, it comes through this peculiar route. And there were other examples like that. In every animal, we found weird loops like this. It was as if the whole thing was not optimized. In an insect or in a worm, in a highly evolved animal like that has been around a long time and evolved quite a lot, you don't see these kinds of roughness and changes from one animal to another. But in mammals, we see this all the time in muscle, and that's, I think, telling us something. Especially because when we look at these wiring diagrams in development, we see that they are fundamentally different. They're different in the sense that every single neuromuscular junction that is singly innervated by one axon in the adult is multiply innervated in babies. And this is just a cartoon to say what we've found, which is that in young animals, the motor neurons in the spinal cord, which are shown as those balloons at the top, co-innervate neuromuscular junctions such that every muscle fiber is innervated in many muscles by every axon, and every axon projects to every muscle fiber, whereas at the end of development, you have a much sparser situation where each muscle fiber is owned by one axon. And if this is a metaphor for the central nervous system, it might mean that early on, when a baby can do nothing, remember it takes about a year to walk and 18 years to leave the nest, so there's a lot of development going on postnatally. During that period of time, rather than making wiring diagrams, we may be trimming away all but a small subset of what we could have been. And that's a kind of destructive view of what education does to our brains. And my children are absolutely convinced this is true because they say I am unable to learn anything new at my advanced age. When we look at a single neuromuscular junction at birth, which unlike that single innervation in adults, using a technique that I'll talk about just in a moment, serial electron microscopy, one can trace through all the axons that innervate one neuromuscular junction. So that's a block of tissue where we've cut and looked in the EM, and then Juan Carlos Tapia colored in the muscle green and the axons co-innervating that one neuromuscular junction, each a different color. And there's clearly more than one axon there. And if you look carefully, they seem to be quite happy. They're kind of like an orgy of rolling over each other. They're on top of each other in a very nice way. And here is an example. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 axons at birth, and one of them is, the last one is just already about to leave. So we've done this to many neuromuscular junctions, and we find 10 or more axons at each neuromuscular junction at birth. Therefore, greater than 90% of the synaptic branches that in a mouse now that are present at birth get eliminated earlier, sooner or later. And one might imagine, could this be true in the central nervous system as well? We have been trying to understand why, and I'm going to make short work of a very long project, which was to understand if we can figure out why some axon wins and all the others go away. And to do this, we did the same kind of wiring diagram I showed you in the adult, now back in development when neuromuscular junctions are multiply innervated by more than one axon. We didn't go back to birth, but at 1 week of age, some neuromuscular junctions like this one are multiply innervated and others are singly innervated. And then by serial reconstruction, in this case with confocal, one can find the identity of every axon at every neuromuscular junction in a muscle and get the whole connectional map, the whole connectome in a baby muscle. I'm going to show you one example from Ryan Draft's work. He's a graduate student who did this. This is a muscle that had 6 different axons innervating it. And the most common thing he saw in this developmental stage of multiple innervation was that the purple neuron and the red neuron shared 66 neuromuscular junctions. In this particular muscle, the next most common thing he saw was 61 muscle fibers shared by those two, 60 by those two, 54 by those two, 50 by those two, making you think that there should be a substantial number between the red and the one at the bottom, perhaps. But in fact, there was only one shared by those two. The next most common actually was 29 by those two, which actually skips one in the particular order I've shown here. 22 neuromuscular junctions shared by those two axons, which skips one. 21 by those two, which also skips one. 18 by those two, which also skip one. 16 neuromuscular junctions by those two axons, which skips 2 in this order. 7 by those two, which also skips 2. 6 by those two, which also skips 2. 4 by those two, if you go around clockwise, skips 3, and then finally that 1, where you have to go around the horn all the way. So this is a big mess, no doubt about it, but there's something going on here. And we have been working hard at decoding this, and finally, I think, figured out what was going on, which is if you look at any one of these axons, you find they have a graded preference about who they want to remain multiply innervating with. This neuron, the red one, has 66 connections with the one on top, and only one with the one at the bottom. So we just replotted this to show a distance, that neurons that are closely related should be near each other and neurons that are farther related should be far apart, like this. Now this is not a topography. This is just the connectivity map that's now been abstracted to space, and you can see that red and purple maintain 66 connections at this moment in time, but red and violet at the opposite extremes only have one connection. So that's for the red neuron. If we do the same for the purple neuron, and see red, purple, salmon, orchid, green, violet, that order, I've left it red, purple, salmon, orchid, green, violet. Again, purple fits the same scheme. It's nearest neighbors. It has the strongest co-innervation with, weaker with orchid, less so with green, and nothing with violet. Salmon also is closely related to purple and orchid, but less so with green and red, and a little bit with violet. And the same is true for the orchid neuron, for the green neuron, and the violet neuron, except for there's one missing connection. Violet and purple should have something, and we didn't see anything, but violet and red had one, and that could be in the realm of an error of our measurement. So here it is all together. There's some kind of linear order connectional matrix that has nothing to do with space in the muscle. And when we looked at other muscles, we found the same general principle applied everywhere. And this was very interesting. We weren't expecting this, but there is a motif. There is a cellular organization in the middle of the period of synaptic reorganization. To make a long story very short, we've done physiology now, and we are convinced that this order is actually the recruitment order of neurons that are activated in a particular order in development. When a nerve cell is active, it causes muscle fiber twitching, and when you want more twitching in a muscle, you add additional neurons firing on top of the first. So in this muscle, the red neuron fires first, followed by the purple neuron, followed by the salmon neuron, followed by the orchid neuron, followed by the green neuron, followed by that last color, which I can't remember what it was. This is Hedeman's size principle, and what's the big deal here? Well, once you know the relation between the wiring diagram and the function, you can say that the wiring diagram per se, its structure, reveals the experience that set it up. Probably that neurons that are firing next to each other in this recruitment order are remaining connected to each other more long than neurons whose firing patterns are very different. And that means we've decoded a wiring diagram so that now when we look at the wiring diagram, we can infer the experience that set it up. And that indeed would be engrammatization, although in a part of the muscle, a part of the body that none of you care about, which is muscle. You all want to know about the cerebral cortex. So when we try to do the same thing in the cerebral cortex, this is the Technicolor Golgi. It looked promising, but when we zoom in and try to get all the wires, big surprise, there's just way too many wires. It's way too dense to figure out. So we've taken two strategies here. The first is to only label subsets of wires of a particular class using a new version of Brainbow, which is a virus. We can use it in animals where only inhibitory axons, for example, express Brainbow. And this is an example of an animal where all the PV positive inhibitory neurons express Brainbow, but nothing else. And these big dark blobs in the middle that are completely covered with these colored boutons, those are the pyramidal neurons that are strongly inhibitorily innervated. And with these colors, we can now reconstruct, and Luke Bogart is doing this work in Takao Hensch's lab, reconstruct the wiring diagram of inhibitory connections. Some of the inhibitory neurons you can see an outline near the top. There's a green one at the very top, and now there's a red one, and they're not covered with inhibitory interneurons, but the excitatory neurons are. So that's one way. But, you know, what if you really want to see all the connections in the cerebral cortex? The approach we've taken is to cut the brain very thin with a weird machine that looks like a movie projector, but actually is collecting sections of brain on tape. I'll just zoom up on what this is. So we're cutting a brain in plastic. The brain sections float on water, and then a conveyor belt in that water bath picks them up one after another. We then take those sections on tape. We linearize the brain, basically, into a bunch of sections. We're cutting typically at 30 nanometers, at very large sections, and we can do about 1,000 sections a day. We cut those sections and we put them onto a wafer that's very flat, and then we put all the wafers together into a library. So now we have those wafers are about the size of a CD. So from that we can have a large piece of brain all sectioned. And then that's what one of those wafers looks like. Has about 180 sections. There's 55 wafers of a piece of brain, 10,000 sections. And then we take those wafers and we image them in the electron microscope. Bobby Kasturi is holding his hand very still, so you can see what these sections actually look like. So that is a section of cerebral cortex. It looks like the state of Maine, but it isn't. It's a cerebral cortex and the hippocampus at the bottom. The big, large white things are blood vessels, and then the white spots are nerve cells. The white streaks you're seeing are apical dendrites. And then you start seeing myelinated axons, and then as we zoom up further, eventually you will see right in the middle a synaptic terminal making a synapse, an axon making a synapse on a dendritic spine and a spine apparatus. Of course, that's one section, and you could do the same thing section after section. And here's a movie of 2,000 of these sections at low resolution. We take these at 3 nanometers. This is shown at 30 nanometers. I can't show the whole data set at 3 nanometers. The screen is not big enough. Those big circular things in the middle, for example, that's a neuron. These big things moving around are either myelinated axons or big apical dendrites as we cut through this volume section by section. If you zoom up higher, you see it's just neuropil. There's huge numbers of synapses there. A neuron cell body is at the very top to give you a sense of scale. And if you look with your eyes, it's very easy to trace these objects from section to section. In fact, any child with a crayon, if you gave them a coloring book with 2,000 pages, could probably do this for you. And indeed, Daniel Berger in Sebastian Sung's lab made for us a nice computer program that allows us to color in, and we have lots of people now coloring in brains this way. So this is a hand colored in segmentation of 216 cubic microns. It took him 2 months to do that coloring in. But after the coloring in, you can then just take that and re-image it, which is the same data now. The big dendrites first, and then I'll put in all the little axons. And so there you have a little piece of brain, and this is totally useless information. It's useless because every side of this block orphans this material. We don't know where it came from or where it's going. To make matters worse, that's that piece that he did, that colored box in that image, and that image sits there in that piece, which sits there in this section. I don't know if you can see a little green dot somewhere around here. I can't see it from where I'm standing, but somewhere around here there's a green dot. That's bigger. That's the smallest pixel I can show on the screen, bigger than what's been done. To give you a sense of what an Fmri voxel is at this scale, that's an Fmri voxel. A word to the wise. When we began 3 years ago, we were just trying to figure out how long it would take to do a cubic millimeter. We were going 0.5 million pixels per second, so a cubic millimeter would take 2.24 centuries. I could get no graduate students interested in this project. And then we've been making improvements, and now we're going 20 million pixels per second. We're generating a terabyte a day, and in 5.6 years, we could have a cubic millimeter. In a month or so, we're going to double that, going 40 million pixels per second. So we would have a cubic millimeter in 2.8 years. And in a few years, we're going to get a very esoteric multi-beam scanning electron microscope, and that's going to let us go about 1 billion or more pixels per second, and then a cubic millimeter in 3 weeks. So what happens with this kind of data? First of all, you have to color it in, and the hand coloring in of that little box took 2 months. We now have, thanks to Hans-Peter Pfister, an engineer at Harvard, a really remarkably robust automatic segmentation algorithm where it's all colored in. This is about 6,000 Daniel Berger units, if you will. So it's a much faster than a human being. It needs editing. Humans have to correct it, but you can see it does a pretty good job, except on the Golgi apparatus in cell bodies, which are very psychedelic because it doesn't really know what to do with them. But except for that, it's pretty good. And I'm going to end by showing you basically what it looks like when you go in and try to saturate reconstruction of a small region of brain. It's much bigger than anything anyone's done before in cortex, but it's 600 cubic microns, where we've tried to get every single element around the apical dendrites of 3 adjacent pyramidal cells. Those are the cells, and that's the region that we saturated. We've made lower scale imaging of this whole area, but that little area where you see that nest of white is where we've labeled everything, every glial process, every dendrite, every axon. And here are the axons that innervate those 3 dendrites in that little region. It's shocking. I mean, it's just disturbing when you realize this is still a minuscule part of those cells, and already there's a massive amount of innervation one has to deal with. If you pull out some interesting ones, you find that more than half of these axons make multiple synapses on the same dendrite. And these are a couple of them that make, and not adjacent dendritic spines, but many spines distributed over space are innervated by these axons. Finally, because I'm at an undergraduate campus, and every undergrad who wants to go to medical school is willing to do anything for a letter of recommendation, we have asked these students to find every synaptic vesicle in every axon. And we're in the middle of that, but I'll just show you progress at this point. So these yellow dots you're seeing here are the synaptic vesicles. This is not an artist's rendering. This is actually the data of the synaptic vesicles in a bunch of synapses. So each of these boutons here is a letter of recommendation. And here is just another view of this. So I have my work cut out for me. But, you know, this is the level of resolution one may need if one wants to understand really the proximate causes of psychiatric illnesses. And, you know, we're just starting, but this is where things are now. So I just want to tell you this is obviously work of a lot of people. There's a team doing the rainbow work, a team working on the electrons. I have many collaborators. This is the kind of project that no lab, I believe, can do by itself, and we have collaborators up the kazoo and necessary, and support from a number of places. So thank you very much for your interest.
I told them it would be awe-inspiring, and that was really an amazing presentation. We have time for a few minutes of questions, so if people would like to approach the microphone in order to ask a question, please do so.
Hi, Jeff. I was wondering if you could comment, now looking into the future, since you have definition in place, about how one might study variation in connectomes and its relationship to disease. So if you were to draw the analogy with genetics, for example, being able to analyze a lot of genomes, look at common and rare variants is very important in understanding which variations are part of the normal spectrum and which ones are associated with the disorder. So I wonder if you could speculate a little bit about what it would take to get to this point, or if there's any reason to believe how stereotypical or variable connectomes might be to be able to make those kinds of associations.
Yeah, I think this is a great question. I think in mammals, as I said, looking in muscle, there's a remarkable amount of variability from one wiring diagram to another. However, that shouldn't be, one doesn't, at least I don't throw up my hands and say, oh, it's hopeless. I say it's sort of like every chess game is different from every other chess game. Once you understand the rules of chess, those variabilities all fit within the same rubric. They're rules that underlie that variation. And I suspect it will be the same with connectomes. There will be normal variation and then pathology. And the pathology, I suspect, will look quite different from the range of normal variation. And indeed, in muscle, that's what we saw. That linear connectional matrix was different in every muscle we looked at, but they all showed the same rule. If we found a muscle where there wasn't a linear connectional matrix, we would say that's aberrant. Even though every wiring diagram instantiation was different, there were certain rules that obtained. So I'm hoping the brain will be the same way.
And I'm wondering if the same applies to thinking about changes in synaptic connectivity with experience, which is kind of where you started your lecture.
Yeah, so my view is that evolution has really not had a lot of animals that learn something in early life and live for years and hold on to that information for years. Mammals do it and birds do it. I'm not sure what other terrestrial vertebrates do it, but not too many. My suspicion is that there aren't 10 million rules that govern that kind of learning. And I think information, whether it goes in through your ears or through your eyes, once it gets to the cortex, it's just action potentials. And so I think the rules that make you recognize a sound or recognize a visual issue will probably all be the same kind of rules. So I don't think there's going to be a lot. It's hard, but I don't think it's hopeless to imagine that we will come up with some kind of rules, not necessarily me, but eventually when connectomes are kind of more routine, seeing how wiring diagrams based on experience are organized, just as we did in muscle, which is a toy problem, to be sure.
You showed us very elegant rules in a simple system out in the muscle, and then you jump to a very, very complex system, which is the heart of the cortical chunk. Have you looked at all at more simple CNS things, like I shudder to say that it's simple, but say dentate innervation to CA3, to see if there are rules in that one particular synapse that correspond or that are in some way make unifying a statement like you saw in the muscle synapse?
The place we've gone next, and we're working on right now, is neurons in the peripheral nervous system, as opposed to muscle fibers. I think it's a fair argument that many people in this audience would probably say is that muscle has nothing to do with learning and memory. I think muscles are honorary neurons because they fire action potentials, they have nicotinic receptors, but most people deny that. But there are neurons in the periphery that also undergo really remarkable changes. That's one place we're looking. The other place is the cerebellum, which is very reiterated, but also undergoes a remarkable rewiring and development when climbing fibers retract branches. The dentate we've not done anything with. We have a little bit of reconstruction there, but again, we don't see enough. You have to do too big a volume there to make sense of it. So that's not where we're doing anything yet.
I'm afraid we have time just for one more question.
Okay. Well, thank you. It was a great talk. Sort of hard on people who are red-green colorblind, but otherwise very cool.
I am so sorry.
There's nothing you can do about it. My question is, this society, pharmacology, big pharma, most of psychiatry has been obsessed with neurotransmitters, like for as long as I've been hanging out with this crowd. And how do the neurotransmitters fit into this?
Yeah, this is a great question. So, because we don't have a lot of time, I'm just going to say that if one just does connectomics like this and ignores the revolution in probes we have now for neurotransmitters, peptides, and the like, one really is going to throw away a lot of potentially good information. So ultimately, one has to have is a connectome with, frankly, the proteome superimposed on top of it. Not just one neurotransmitter, but all of them. And that's what we're working on now, how to superimpose molecular information of all the neurotransmitter types right on top of this.